Welcome back to Vitamins in Biochemistry. My name is Kevin Tokoph. Make sure to like this video and subscribe to the channel for future videos and notifications. Alright, in this video we're going to go over vitamin A structure, metabolism, and how it works in the human body. But first we need to start with plants. And actually we've been doing a very large study of plants, and so plants produce molecules called carotenoids. Alright, when we talked about photosynthesis in the photosynthesis playlist, we mentioned that the plant has a lot of light-absorbing pigments that it uses to absorb various wavelengths of light. The most obvious ones that we talked about were chlorophylls, and there's different types of those, but there are also what we call accessory pigments called carotenoids. All right? You probably have heard about those in the context of a botany class or maybe general biology, but carotenoids are accessory pigments that absorb other wavelengths of light more for the purpose of photoprotection from excess energy from the sun. Okay, they're a protective mechanism that the plant uses. And carotenoids come in a lot of varieties, and these are 6 that are shown here, and there are lots of others. But it turns out that when we eat plants, so maybe we eat things like orange plants, because most of these emit orange, so things like pumpkin, or orange peppers, red peppers, even yellow peppers, things like sweet potatoes, things in that general color range, they probably have carotenoids. And you've probably heard of beta-carotene. And the reason you've heard of beta-carotene is because beta-carotene has what we call the highest vitamin A activity. What does all that mean? You're going too fast. Some carotenoids, and we'll talk about which ones, can be converted in humans when we eat them to vitamin A. I'll show you the structure of vitamin A on the next slide, but suffice to say some carotenoids can be converted to vitamin A and some cannot. The ones that cannot, we would say, have no pro-vitamin A activity. The ones that do or can be converted to vitamin A, we'll say have pro-vitamin A activity. Okay? And certain ones have more vitamin A activity than others. Okay? The highest degree of vitamin A activity is in beta-carotene. And this is the reason why. To analyze just structurally what might have vitamin A activity and what might not, is in general I'm looking at this ring right here. So these rings on the very ends. As long as the ring looks exactly like this, now I can flip it upside down if I need to, but as long as it looks like this ring right there where my mouse is, it'll have vitamin A activity. So if I look at alpha carotene up above, this ring looks identical to that one. Same methyl groups, these methyl groups right here, they're exactly the same. Those two look exactly the same. So I would say alpha carotene has vitamin A activity. Now if I look at this side of beta carotene, okay, this ring is exactly the same as this. You have to sort of imagine flipping it upside down, but you would see that this ring is exactly the same. It's just the reverse of what's over here. It's a mirror image basically, but it's the same ring. Now if I go up to alpha carotene, is that ring the same? And the answer is no, the double bond is in the wrong place. So because of that, alpha carotene would have almost a half of the vitamin A activity as beta carotene, because beta carotene has double the vitamin A. Okay? It has this ring right here and this one, whereas alpha carotene only has one. There's another carotene that we could talk about called gamma carotene, which is not shown here, but gamma carotene also has one of these rings. So it's also going to have close to the activity of alpha carotene, but only about half of that of beta carotene. Can you see any others on here that might have some vitamin A activity? Well, lutein is not going to, because these are hydroxylated. Zeaxanthin is not going to. What about beta-cryptoxanthin? This one has a hydroxyl group, so this one's not, but this ring right here is. This one looks exactly like it does in beta-carotene, so this one will actually have some vitamin A activity. Would lycopene? No, because the rings aren't closed. Okay? So it turns out there's really only 4 main carotenoids that have vitamin A activity. It's going to be alpha, beta, and gamma carotene, and then beta-cryptoxanthin. Okay? And another thing they do in plants is they absorb excess UV radiation to prevent oxidative damage due to free radicals. That's what we talked about, the photoprotection. They protect plants from excess light. But we don't use these as carotenoids. We actually have to convert them to something useful. Okay? I'm going to focus on this pathway over going to the right. Turns out there is beta-carotene, and what we're going to have to do is use an enzyme called carotenoid monooxygenase, or CMO. and there's two types. But carotenoid or carotene monooxygenase is basically going to split the beta-carotene in half, right down the middle, in the middle of this double bond right there, and essentially it's going to form two molecules, if it's beta-carotene, of retinaldehyde. If it was any one of these others, alpha or gamma-carotene or beta-cryptoxanthin, reaction of those carotenoids with carotenoid monooxygenase would only give one retinaldehyde. But because beta-carotene is symmetric, beta-carotene reaction with this enzyme actually gives us two retinaldehydes, or retinal. And that's why beta-carotene has the highest vitamin A activity, is because it produces more retinal. So this is written as retinaldehyde. It's also called retinal. Okay? Retinal can be converted to retinol by a simple dehydrogenation reaction, and then that retinol can actually be converted to an ester by an esterification enzyme. Okay? The retinal can also be oxidized into retinoic acid, which plays some roles in some biosignaling events. So basically all we're doing is we're altering the oxidation state of the aldehyde, either oxidizing it into an acid or reducing it into an alcohol, and then we can esterify that. The reason we care about that is because it's for the function of retinoids in humans. It turns out that retinoids play a huge role in vision in mammals and other organisms too. So what I'm going to do is start off by talking about this form of the retinal, and that's 11-cis retinal. So 11-cis retinal is called that because normally when we looked at these, all these double bonds were trans. It turns out that to be functional, initially, the retinal has to have a cis bond right there where my mouse is. And so you call it 11-cis retinal. We're then going to transport it into the rod outer segment, which is a cell, okay, of the rod. And the aldehyde right here at the end, thus the name al, retinal, is going to react with a lysine residue of a protein called rhodopsin. Rhodopsin is going to ligate itself to the 11-cis retinal, and when it's ligated to this lysine, so you see the nitrogen, it's going to get a new name, 11-cis retinylidene. Okay? This addition of this essential cofactor of rhodopsin makes rhodopsin active. Rhodopsin is not active without retinal. And when you attach 11-cis retinal, it becomes 11-cis retinylidene. Now, when we see, it's due to light. So this H nu right here, or some people call it HV, is light. And the light actually causes a spontaneous isomerization of the retinal cofactor to make the cis bond trans. And so as a result, you call it all-trans retinylidene. Now, that reaction completely causes every double bond to become trans in the retinal, but that also makes this cofactor useless. So the cofactor has to be recycled. Essentially what's going to happen is it's going to be hydrolyzed off by an enzyme. Okay? So now we don't have the retinal attached to the rhodopsin, we have all trans retinal that's floating free. Then we have all-trans retinal dehydrogenase, which is going to reduce the retinal into retinol, all-trans retinol. That all-trans retinol is going to be transported into an epithelial cell, which is the retinal pigment epithelium, and then that all-trans retinol is going to be esterified to an ester right here, esterified to an acyl group, making an all-trans retinyl ester. These are just the reactions that we showed right here, but now we're looking at the physiological significance. This all-trans retinyl ester will then react with an enzyme labeled here as RPE65, which stands for retinoid isomerohydrolase. We're not really concerned much with what it does, but suffice it to say, it ultimately converts this all-trans retinyl ester into 11-cis retinol. Okay, the 11-cis retinol then is going to react with 11-cis retinol dehydrogenase and get back to 11-cis retinal. And then that retinal is going to go back into the rod outer segment where it's going to get ligated to rhodopsin, and you're just going to do this cycle. Okay, this is called the retinoid cycle in humans. Okay, so this vitamin A that we intake, or the carotenoids, get converted to vitamin A, which is retinal. Retinal is the active form in humans, and it's going to be used as a cofactor for proteins in your eye, and that's going to allow you to see. This occurs in the rods for the protein rhodopsin, and it's also going to occur in the proteins in the cones, which help you see color. Okay, a similar process is going to occur. The same cycle. So hopefully that gives you an intuitive sense more on the biochemical level of why vitamin A's are important. So thank you for watching this video. I hope it made sense. Make sure to like it and subscribe to the channel for future videos and notifications. Thank you.